Let me just tell you, I fail 85% of the time easily. So just that failure is a part of the process, and failure brings you closer to success. So never be scared to take the risks and to fail, because that just takes you one step closer to the next big thing.
Welcome to Longevity by Design, a podcast designed to give individuals access to the leading scientific information in the field of longevity. The ability to add years to your life and life to your years needs no opinion. Join us as we ask science to take the wheel. In each episode, Dr. Gil Blander joins a co-host and an industry expert in the field of longevity, shining a light and getting the answers to the key question: how can we live a longer, healthier life?
Hello, I'm Dr. Gil Blander. Welcome to Longevity by Design, how to live a longer, healthier life. We are produced by InstaTracker, your science-based guide to optimize your body from the inside out. Today's guest is Dr. Louise Hecker, Associate Professor of Medicine at Baylor College of Medicine and Research Scientist at the Department of Veteran Affairs. A recent work published in NPJ Aging explored the systematic effect of psilocybin on aging biology, including cellular lifespan and survival in aged mice, raising a provocative question about novel pathway to improve health span. Louise, welcome to Longevity by Design.
Thank you. Thanks so much for having me.
No, it's exciting. And before we'll dive in, if you, the audience, enjoying Longevity by Design, please follow and leave a review on Apple Podcasts, Spotify, and YouTube. It's help others to find this science-first conversation. Louise, before we go to the exciting result that you got about psilocybin, can you walk us through your scientific journey and how your work in pulmonary fibrosis and tissue repair led to the broader question about aging?
Yeah, it wasn't a straight line, I can tell you that. My background in training, starting from my undergraduate research, was always in repair and regeneration. I've studied all types of systems, from amphibians to tissue engineering models, bioengineered organs. And when I started in my postdoc, and one of the things I learned actually along the way was that I used to just pick projects. I used to pick the projects that were what I gravitated toward. And over time, I started really picking mentors. And when I got to my postdoc, I picked a mentor I really wanted to work with. And he happened to be studying aging. And he felt like this was the perfect match for me, because he said, having this background in repair and regeneration, this is perfectly suiting to you for studying aging, where repair and regeneration are less efficient. And so that's when I started aging research in my postdoc, and the whole of my professional career since has been really focused on age-related diseases, fibrosis being one of those things. We don't typically see diseases of fibrosis unless you're elderly. And so understanding why that is and what goes awry in the context of injury repair and aging, that's really what most of my career has focused on.
Yeah, and that's led me to the question, why does regeneration fail as we get older?
Yeah, that's really just how we've evolved and how we're designed. And aging is a natural physiological process of deteriorating physiological function that is normal. And of course, we would all like to live healthier longer. That's the goal. And so understanding how to do that, and that's really what I've been focused on, is how can we help these patients that are otherwise healthy not get taken down in the prime of their life by an age-related disease, and understanding what happens, what goes awry, what are these age-related defects that happen, and how can we correct them?
Yeah, no, that's a big question. And unfortunately, we won't have time to discuss it too much today, because you recently published a very exciting paper, amazingly exciting for me, I would say, in the journal NPJ Aging around 6 months ago, basically exploring the effects of psilocybin on cellular and the organism lifespan and survival. So let's start by what motivated you to do this research?
Yeah, it's a good story, actually, which started with just a friend of mine that just was always asking me questions about psilocybin. And when you're a scientist I think a lot of your non-scientist friends think you know everything about science, and really we don't. We know what our scientific area is, and non-scientists think scientists know all things science. And so he was always asking me actually very good questions about psilocybin. My friend Aaron, who we used to work out together 3-4 times a week. And so I never had any good answers for him because I just didn't know anything about psilocybin. And one day I just decided to start reading on PubMed. And he gets his information from Netflix and podcasts and social media. Not that's a bad way to get information, but as a scientist, I wanted to really understand what was known, what is actually scientifically known about psilocybin. And that's when I started reading and just went down a rabbit hole. I thought I was going to be reading for 15 minutes, and 4 hours later, I was still reading. And I just couldn't get enough of this topic. I was so fascinated. Psilocybin is an interesting drug, and I had no idea how much work had been going on in that area. There have been nearly 200 clinical trials. It's being used for so many different disease indications for all types of psychiatric conditions, anxiety, depression, PTSD, bipolar disorder, and a number of other psychiatric neurodegenerative diseases, Alzheimer's, Parkinson's, and many other things, anorexia, chronic pain. And that was the first question I had is, how is this one drug working for so many disease indications? And then the other big question I had from reading is people were reporting that just one dose of psilocybin was leading to positive impacts 5 years later. And I just thought that was astonishing. How does that work? And I couldn't wrap my brain around how does that work. And someone had put this idea out there that maybe the way that it's having these long-term impacts is that it's having impact on aging. And that can lead to a number of different impacts, right? That could lead to these lasting effects. And so I thought, I study aging and I can test that. And that's really where everything started. So I had zero background knowledge or experience working with psychedelics or psilocybin prior to embarking on this project. But I'm just a curious person, and I like trying new things, and I like, exciting ideas. I'm a very adventurous, I guess, scientist. And so I thought, let's see if there's something to this idea. And that's kind of where it started.
Yeah, that's an amazing example of how a discussion, I would call it a corridor or water cooler discussion with a friend, change a complete career of a scientist. So that's an amazing example, and it's good to show that for our audience, if you have a friend scientist, talk with him, ask him, challenge him or her, and try to see maybe it's they will be so curious what Louise did that basically would change their career completely. Now, my background for some of our, let's say, geek audience that don't know what is psilocybin. Can you explain what is it?
Yes. So psilocybin is the active compound in psychedelic mushrooms. And people most recognize psychedelic mushrooms, of course, because it has psychedelic effects. 99% of what we know about psilocybin come from what it does in the brain, what we know scientifically about what it does in the brain, and what we know from clinical outcomes. In addition to, as I'm sure you're aware, there are tons of anecdotal stories out there about psilocybin. And so there's a lot of information, anecdotal information, and very little scientific information, actually.
I assume that most of our audience will attach them to basically taking recreational mushroom to feel high and things like that. That's the most use of that, correct?
I don't know about most use because it's certainly being used, of course, in clinical trials. It's a Schedule I drug, which means it's the same class of drugs as heroin. So it's illegal recreationally, except for it's been decriminalized or legalized in some cities and states. I know Oregon was the first state to decriminalize it. So there are a growing number of states and cities that are making it possible for people to gain access to it, and certainly people are taking it for different reasons, for therapeutic uses as well as, I'm sure, recreational uses. The reason it became a Schedule I drug is because of In the 70s, it became a popular recreational drug, which unfortunately led to these government sanctioned regulations designating it as a Schedule 1 drug because of its potential for addiction, lack of accepted medical use, and lack of accepted safety profile are reasons why something is classified as Schedule 1 drug. That led to a really sharp decline in psilocybin research and funding at the time. And more recently, there's been a resurgence of interest in psilocybin research. Certainly, the clinical evidence coming out has really strengthened the argument that, for example, its potential for addiction is one of the reasons it's a Schedule 1 drug, yet it's being used actually to treat addiction. The same with the safety profile. It's been in so many clinical trials, and it's been designated as a breakthrough therapy by the FDA, which speaks to its safety profile being very good. So these things are changing, and hopefully we'll see changes in reclassification of its schedule status. That would reduce some of the barriers that people like me face when we're trying to study psilocybin.
Yeah, so let's talk about that. So you had a discussion with your friend, and you came with the idea, let's see what is the effect of it on aging, longevity. I assume that you need some funding, you need to understand what is the dose to use, what kind of animal model, what kind of cells model. Can you get us into those decisions and how you cross those murky water to do a study in human cells and in nice animal models?
Sure, yeah. The first thing that I thought of doing was what I thought was a very simple experiment, which was just we use as aging scientists models of cellular lifespan, models of what we call replicative senescence. Cells have a replicative lifespan, and it's very reproducible. And just like people, cells only live so long. And so you can watch that cellular lifespan and quantify it with or without a treatment. And so the first experiment was really simple. Let's watch cellular lifespan in human cells with and without psilocybin. And so I thought, this is easy. We do these types of experiments all the time. And I went in the lab and I said, let's order some psilocybin. And that's where I realized, okay, this is going to be tougher than I thought, because you can't just order psilocybin. You need a DEA license. You need to have DEA inspections of your lab. This is a very long process that requires a lot of paperwork and time and effort, and just to bring the substance into the lab. And
so one comment about that, I remember when I came to MIT as a postdoctoral fellow, I wanted to do some deacetylation experiment with radioactive ethyl group. And I ordered from the catalog like a big amount of radioactive acetyl. And then the MIT police came to me and said, what are you going to do? Are you going to contaminate Cambridge? So it's the same like that. I assume that something ring the bell and you couldn't. So how did you cross that river?
Yeah, I was like, so actually the only psilocybin that we're able to order is like basically not pure psilocybin, but essentially like a psilocybin that you can use to spike a drug test so that like it's your positive control. So it's not pure psilocybin. So we can't use that for experiments because it's not a pure form of it. So it was a long process, probably about a year.
Wow.
Before, from the time that I had this idea and went into the lab to order it to the time I actually initiated an experiment was probably about a year. That's probably about how long it took to go through this process of getting a DEA license and all the inspections and all the regulatory things that come along with that, and the ability to possess it in the lab. So a year goes by and I'm just, this is like a side project. And when it comes, and we'll just push this along. And then the psilocybin, I remember actually we have a picture of all of us the day that it came. We all took a picture with the psilocybin bottle. Yeah, we got it. So it was an exciting day. And we didn't know what was going to happen. I just thought, this is another one of my crazy ideas. And trust me, I've tested a lot of things that are crazy that don't work. So I don't really go into these things with an expectation other than I thought this was an interesting concept. It was intriguing enough to spend the time and energy to want to try to test this. But I just let the data speak for itself. And then what we found was that in a dose-dependent way, meaning the more cells. Actually, let me back up. When we give to cells, we give psilocin, which is the metabolized form of psilocybin. So when we ingest mushrooms, psychedelic mushrooms, through the digestion process, it's broken down into psilocin, which is the active metabolite. And that's what your cells see systemically. So in cells, cell experiments, we use psilocin, the active metabolite. And what we saw is that in a dose-dependent way, psilocin led to this dramatic increase in cellular life extension. So cells lived much longer in a dose-dependent way. And that was the first major result.
Yeah. And can you quantify it? Like if, let's say, a normal fibroblast that you use, replicate 25 or 30 replication, what is the increase in the number of replication and percentage, more or less?
Sure, I think, and we did initially, in what's in the paper, we did these in fibroblast cells. We did it in lung fibroblast cells because I happen to study lung fibrosis, and those are a key cell type. So I just happen to have those, and they're a very popular model for replicative senescence that we've used. And so I just started with the model that I had. And then we saw some differences, like I think maybe like 30, I'd have to look back in the paper, geez, up to 100 micromolar, you could get 50% extension of cellular life extension. And at the lower doses, you're starting maybe 30 micromolar, you see like 30% life extension. And then we repeated these studies with skin fibroblasts. So we wanted to see, okay, when a different cell type, does it do the same thing? And results were essentially identical.
Yeah, and as a fellow scientist that is not involved in this research, I have to say that's very impressive and significant to see such an effect with one compound. That suggests that there is something real there. And,
right. We were mind blown. The first thing that we thought, because the cells kept dividing, so we thought, was this cancerous? Because you think more cell division, that's a risk factor. But that's not what we saw, at least in the cells, because cells did eventually. Cancer cells, by definition, are uncontrolled cell death, so they don't stop dividing. And that's not what we saw. We just saw that cells continued to divide. They just divided longer and they lived longer. But eventually they did grow old. Just it took them much longer to get there. So that was the first major discovery. And from there, that's when we started looking at what's happening inside the cells at the biochemical level and looking at different hallmarks of aging. That's where we started next.
Okay, and what have you seen? What are the changes that happen when you treat the cells with psilocybin?
Yeah, I still remember the very first kind of batch of results that my technician Yoon brought to me. I basically said, here's every aging marker that we have in the lab, run it. And so what we saw is that we compare them to young cells to old cells. And so we see typically certain markers of aging, like cell proliferation goes down, cell cycle arrest markers go up, DNA repair goes down, things like that. So we saw that normal sort of markers of aging that we see in the vehicle treated cells. And then again, in a dose dependent way, every single marker that I looked at was rescued to some extent in a dose-dependent manner by psilocin. So that was like mind-blowing, because sometimes again, I'm just fishing here, like what's happening with aging markers. And when you see every single marker being rescued, I was just shocked. And so that inspired me to just keep digging. And so we looked at things like oxidative stress. So oxidative stress levels were dramatically reduced. And we found that maybe in part due to the fact that psilocin leads to decreased oxidant production as well as increased antioxidant responses. And so typically oxidative stress accumulates with age, and the psilocin treated cells had oxidative stress levels that were even lower than young cells. So they were, by appearance, phenotypically, the cells were appearing to be behaving like young cells. They had better DNA repair. They had decreased oxidative stress. They had better regulation of their metabolism. They had, another thing is we looked at telomere length. So telomeres important as cells divide. until they shorten to a point where cells can just no longer divide. And telomere length, as I'm sure, is a well-validated hallmark of aging. And so we looked at telomere length in the psilocin-treated cells. And of course, the old cells had, vehicle-treated cells had significantly reduced telomere length, and the psilocin-treated cells had preserved telomere length. So the telomeres were not shortening as rapidly as they normally do as a result of normal aging. So they had better protection of their DNA.
Yeah, it sounds like the treatment hit a lot of the hallmark of aging, and basically it's more, it's not a specific for one pathway, but it's working as much as I understand from what you're saying on many of those. Have you seen any pathway that it's not affected by the treatment?
Yeah, that's a good question. I think one of the things that we haven't published yet, but we're working on, is doing all of the omics profiling from all of the different organs from these mice after the treatments. And we're still wrapping our head around and what is unique and what is rescued essentially in the different organs. And so it looks like many of the age-related pathways are rescued, but I will say that we are seeing some differences. So some organs seem to have, psilocybin seems to generate more protection for some organs compared to other organs. So I think we're seeing some organ-specific effects. So I think it may be cell type specific of what the type of protection that you can get from psilocybin. So these are still questions that the study really just opened up 1,000 more questions essentially. And so we're still digging for answers there.
As a Longevity by Design podcast listener, you understand the value of improving your health for today and for all the years ahead. And if you want to live your healthiest, longest life possible, you need to understand what's going on inside. At Inside Tracker, we take a personalized approach to health span optimization that eliminates guesswork from your wellness plan. Inside Tracker analyzes blood biomarker and DNA data, along with physiomarker data from fitness trackers like Oura Ring, to deliver personalized food, supplement, lifestyle, and exercise recommendations that allow you to take control and improve your health span. And for a limited time, Longevity by Design listeners can get 20% off at the Inside Tracker store. So if you're ready to receive a personal health analysis and data-driven wellness plan to optimize your body for the long haul, then it's time to start inside. Visit insidetracker.com/podcast to get started today. That's insidetracker.com/podcast to get started today.
You started to touch on it. Following the experiment in the fibroblast, you moved to, okay, let's test it in mice. So can you describe that experiment? What have you found and so on?
Sure, yeah. So because of the very exciting cell data, we thought this is worth going into mice, seeing if this works in live animals. And that was also challenging. You mentioned earlier about funding. And so it was very difficult to get this project funded. I wasn't successful initially getting any types of federal funding. And with the tiny bit of pilot data that I was generating in the lab, Emory, at the time I was working at Emory University, and they gave me a small pilot fund to look at the cells. And so with that pilot money, that's the cell studies that we did that I just described, turned out so incredibly positive that I was able to get a second round of pilot funding from Emory to test this in mice. And knowing that we didn't have this unlimited amount of funding and that we just had a small amount of pilot funding, we were really focused on, okay, how do we design this experiment? We've got one shot, and what's the best way? So we spent a lot of time really trying to design the best experiment. And keep in mind, no one's ever done this. To my knowledge, there have never been a study with psilocybin in aging. There's never been a study, at least in animals. I mean, it's been given clinically, but very little is known about giving psilocybin in the context of aging. And certainly there's never been a study where you give repetitive psilocybin in aging. There was so much unknown, and that made it challenging to get this study, to design the study, because in some ways we're guessing. And what we did was really just sit back and think. We had a clinician scientist, one of my colleagues, Ali John Zarabi, who's a clinician, and he's led clinical trials with psilocybin. So he was really instrumental in providing some guidance and feedback and understanding how it's being used clinically. And then we also took a look at basically every study that's ever been done in mice and what's known. So we took everything that was known essentially. And from that, we constructed, this is our protocol based on everything we know. This seems like the most appropriate protocol. And what that was is we wanted to give a realistic kind of geroprotective intervention, meaning it's not really realistic if we say, oh, I'm going to give psilocybin from birth and see if you have, that's not a realistic therapeutic avenue that people are going to want to try, right? So that's not feasible. So when we designed our experiment, we thought about how would this actually be used? And so we used mice that were the equivalent of a 60-year-old person. That's really when in your 60s, really when you start seeing a lot of increased incidence of age-related diseases. So we thought we'll start with aged mice. And initially, what I wanted to study was how does this increase life extension, like in the cells? Because that to me is a good surrogate for if you can increase life extension, then it's likely that intervention increases health span. And so that's really what I'm after. I don't want to live forever. I want to live healthier longer. And I was using the idea of life extension, whatever works the best for life extension logically might have the best impact on health span. That said, I wasn't allowed to do that study because of safety concerns. Like I said, there was just nothing known. And it's the equivalent of saying, hey, I'd like to give mice high dose heroin for the rest of their life. The questions were just, woo, what? This might not even be safe. And those are realistic concerns at the time. So we said, okay, we're going to put together a very strict monitoring protocol. And we're going to give these aged mice psilocybin hallucinogenic dose once a month. And what I was allowed to do is test until any group reached median lifespan. So when 50% of any treatment group had died, that signals the end of my experiment. And so that turned out to be 10 months into the experiment. And what we found is that when 50% of the vehicle mice died, 80% of the psilocybin treated mice were still thriving and surviving. Not only did they have this dramatic increase in survival, but the mice phenotypically looked better. They had mice like people also get bald spots, get gray hair, and they were starting to show these aging phenotypes at the start of the experiment. And interestingly, and probably the most surprising result is that psilocybin treatment reversed some of these phenotypes. So bald spots grew back hair, white hair grew back black. On top of the fact that these mice had this dramatic survival improvement, the mice just looked healthier phenotypically, and it's something that everybody noticed in the lab. It's like, wow. So those were some of the main findings from the study itself.
Yeah, and that's amazing. I'm looking at the paper right now. The size of this, the sample size of this study was tiny,
Yes.
around 30 mice in each group.
Right.
And I assume that when you did the sensitivity analysis, there was a high chance that you won't see an effect, even if there is a real effect, because the size is so small.
That's
And
absolutely correct.
And still, again, I'm looking at the survival curve, and it's very significant. So I remember that when I was still in academia, we always said that 1 month in the lab save you 1 hour in the library. And it was a joke. Basically, go to the library 1 hour, and then you will save 1 month in the lab. And that's what you have done. You mine every paper in the literature and design a good study, and then there is a much better chance that the study will work. And I hope that all of us as a scientists will do that, because then we can save a lot of money, but also our time. Instead of doing a lot of experiment, do the right experiment. And yet the results are really exciting and amazing. And now the question is, what next? How do you explain that? And how do you translate those data to real human? Because we know that a lot of studies that have been done in mice are not translatable to humans. So sorry that I asked so many questions, but if you can start by one of them, and we'll continue to the other.
Yeah, I totally agree with you about, an hour in the library. So yes, we really did take the time to look at just absolutely everything out there. And I think that especially with dose, and I think I've seen some people have said this dose is much higher than what's being used clinically and things like that. again, taking so many factors into consideration, like mouse metabolism is 4 times faster than human metabolism. So even taking metabolism into account, but also the goal, again, because we had a limited sample size and limited pilot funding, wasn't to come up with the perfect dose for clinical translation. It was to come up with a dose where we were going to see an effect. And certainly we did. And a lot of, I think, what needs to be done next is answering all the questions that you're asking. How do we translate this to humans? What is the optimal dose for geroprotection? What's the optimal age to start? What if you start at 80? You still have protection? Or what if you started at 40? Would you have twice as much protection? We just don't know the answers to those questions that need to be tested, rigorously tested. We need to think about frequency of dosing. We didn't publish this yet. We're still finalizing the data, but we also did a microdose study. And just to give you a sneak preview, what we're finding is that dose and frequency may do different things and have different benefits. And so understanding the optimal dose for certain benefits, you may need a different dose to have better benefits for skin and hair than you do to have protection in your lungs, per se, let's say. So those are all the questions that we're asking right now. And the only way to know the answer really is to do a lot more rigorous testing.
Yeah. So if you will put it in your priority list, what is the priority 1? What is priority 2? And what can you tell us based on the data that you have that you're still allowed to say?
Yeah. Wow. It's actually quite amazing how, like you said earlier, your life can change from like one finding, because this has just exploded with outside interest, with new questions to ask. And one of the most exciting parts of having this study finally published is just all the people that have reached out to me to collaborate has been so exciting. Certainly things like mechanism, how is it working? That's a tedious process of blocking receptors and doing these tests to see which pathways is this acting, which receptors is it working through. Do you have different impacts when you use different dosing in terms of the pathways that are activated? We're all digging through that. So those are some questions. The question of geroprotection, the optimal protocol, that's a whole separate question that we're trying to attack in different ways. Again, we collected this kind of mountain of data from this. So from the study that's published, we just put the main finding out there, but we've collected from those mice all the organs. We've done epigenetic profiling, metabolomic profiling, transcriptomic profiling, proteomic profiling. And so we have all this data. Now, I'm not a person that does omics research. I always have collaborators. So right now we have a team of collaborators that are working on that data. And hopefully soon we'll have a paper come out with the rest of the findings from that first initial study. So that'll give us some insight into which organs have less protection. Interestingly, the brain, at least according to the transcriptomic profiling, a brain, of course, as you would imagine, has significant rescue of aging pathways, but actually it wasn't the organ with the most rescue. Actually, our transcriptomic data suggests the lungs have the most age-related pathways rescued. So some things were surprising, that psilocybin has such potent impacts on so many other organs systemically that no one's just ever really studied. So this was a really surprising finding. We're also finding that some organs don't have as much protection from psilocybin. So I think understanding how it's working in terms of the different organ systems, certainly along with that, we need to understand potential harms. What are the risks to taking psilocybin? Again, even in clinical studies, I don't know of a clinical study where they did long-term repetitive dosing, certainly not in elderly patients. That needs to be rigorously tested. Are there any risks associated with repetitive long-term dosing? We just don't know. There's never been a study. So that needs to happen. We certainly need to know if psilocybin impacts human aging. And that is something that we can do by making use of clinical trial samples And so I'm working with collaborators to try to develop new studies as well as ancillary studies to existing studies where I would be able to hopefully collect these patient samples and really rigorously characterize and assess the impact on biological aging, which has never been done before. So that'll give us some real insight, at least into how this is working in humans at the doses that are currently being given clinically. So that'll be very interesting and will help to inform the next wave of research.
Yeah, absolutely. And astonished by the result in the mice and the magnitude. And my question is, have you tried to compare the magnitude or the effect of the treated mice, to compare it, for example, to caloric restriction or rapamycin, and see, is it comparable or is it better, or you haven't looked at that yet?
That's like an amazing question that I've thought about so much, actually. And so it's interesting. I've seen actually some scientists on their own, like YouTube or posts or whatever, they've actually overlaid our data over top of other therapeutics. Other scientists are saying this is very promising compared to these other things that are out there. The reason why I think that's tough for me to answer is because it's a little bit like, unless you're testing these things head to head, and even if you're testing them head to head, let's use calorie restriction as an example. That's something that has to happen daily, right?
Yeah.
But how do you really compare something that happens daily, like a daily something, to something that happens in mice, like that you're giving them once a month? And then they have to be starting this treatment at the same age. That's the other thing. And then they have to end at the same age. So it's really tough to make these comparisons. And I think the only real way to do that is to do a side-by-side experiment. And again, even then, because let's say rapamycin, you're going to take that daily. How do you compare a daily treatment to a treatment that you give once a month or once a year? I don't know how you really make that comparison. But yeah, I think what needs to be compared is what's the best protocol out there for any one of those drugs, rapamycin, calorie restriction. Use that, compare that with whatever the best protocol is that we've optimized for psilocybin, and see how and really the end effect is really what is of interest, right? And then if there's this therapy that you could take much less often, that's much more potent, that'd be a great thing if that turns out to be true. We'll see.
Yeah, and also it's much easier to implement caloric restriction. We've seen it. There have been a few human studies, and it's very hard for us to stick to that because you need to basically fast for a few years or maybe more. While here, it's, as you said, if it will stay the same for the mice, if you take it once a month, it's much, much easier.
Right.
Much easier to implement. Absolutely. I think that one suggestion for me is, I'm sure that you heard about the ATP, that they basically testing intervention that extend lifespan in a few different locations. Mm-hmm. So that might be very interesting to see, because then there is always the criticism about mice. It might be only the strain related. It might be only the location. But what they are doing, they are testing it in 3 different locations with a few different mice strains. So I think that it will be very interesting to see what is the result with that. And I think that the only intervention that is not caloric restriction that have been shown to work there was the rapamycin, as much as I remember. So it's very interesting as a next step maybe to do that. And
yeah, I'm so excited to see what comes out of this. I have no doubt this will be validated. As you see in the paper, the results were striking. And so I'm biased because it's my work, but I am super confident that this work will be validated. And even since we've published our paper, like I've seen a couple of studies come out that have started looking at aging in their models with psilocybin, and the results are supportive of our findings as well. And I'm sure, and I can't wait for others to do similar experiments with different strands of mice, with other model systems, and to see what comes out of this. I think it's just a whole new frontier for psychedelic research. When I think of serotonin receptors, and again, we don't know exactly which receptors its impact on aging, but we know that psilocybin acts through the serotonin receptor as one receptor. And when I think of serotonin, initially, and I knew nothing, I don't study this, right? So I just think brain. I think serotonin, I think brain. But actually, I was really surprised to find out that serotonin receptors are on almost all cells. So it makes sense, right, that it's not just going to impact cells in the brain, but it's going to impact all cells that have receptors for that. And yeah, so I didn't actually know that going into studying this, but now that I know that, it makes sense why psilocybin is acting systemically. And I think there's so much more to be learned about what it's doing systemically. We just really have not a lot of scientific data out there. No one's really studied this. It's going to be really exciting what comes next, and all the new research that's going to come out of this finding.
I agree. And a question, a more practical question, or maybe not practical question, is I'm sure that there are a lot of biohacker and longevity driver that would like to live longer, better, and they are now ready to order a lot of mushrooms and I don't know, try to find the right compound. What is your recommendation for them? What should they do?
Oh gosh, yeah, I get asked this question a lot. I'm a scientist, right? So and I'm not a clinician. And I think my position on this is extremely promising data, and science is just, I couldn't be more excited myself. But there just needs to be more research before we start taking this, before I can go out there and tell people, hey, this is something you should be taking. We just need to know more. We need to understand better the dosing. We need to understand better the frequency of dosing. How often should you take it? What kind of doses should you be taking? And we need to understand potential harms. We need to understand if there are any potential negative downsides to taking this long term, because that's what it would require based on my work. We're finding that if you just take it acutely, the acute effects are minimal. It's the long-term effects that are actually. And so how do you dose it so that you have the maximal long-term effects without negative side effects or adverse effects? That's just going to take research and time. So I think people should be optimistic, excited, and keeping their eye on this, but it's just not ready for prime time as an anti-aging therapy until it's been tested a bit more. Now, that said, it's very safe. It's been given in clinical trials, lots of doses higher than what I've been giving in mice. And we just have to continue to study this in not only in cells in animals, but in patients to get a better insight of what will work. And come back in a year. I think you'll be surprised in 1 year, we're going to have a lot more answers.
I 100% agree with you. Wait a year, don't try it yet because the harm might be higher than the benefit.
Sure.
It might not be, but it's very risky. But again, people that want, they can go and buy it. It's out there, so it's up to them. But I agree with you.
I think it's also, to your point, because it's out there and people do have access to it, it's another reason why I think it's so important that we study this more closely.
Yeah.
Traditionally, the government has not funded this as much as the traditional kind of route.
Yeah.
But I'm seeing these trends that are changing. The Department of Veterans Affairs has put out funding announcements for psilocybin specifically. The NIH, I know a colleague of mine just is a part of a $20 million clinical trial that was an award from NIH. So I see the trend changing, but we need to study this more. We need everyone to understand, the government, private donors, foundations, everyone who's watching, to understand that this is a topic that requires more investigation. So it needs to be prioritized and funded. And the more funding that we have to study this, the faster we're going to have answers. So really, it's going to come down to funding and excitement, enthusiasm. I think I think a lot of people are excited, so that I guarantee there's going to be a huge wave of aging psilocybin and or psychedelics research in the next year because of this, kind of opening that door. And I hope that wave continues to grow, and I hope the funding continues to grow, because the more that happens, the quicker we're going to have answers.
Yeah, 1% agree with you. And to finish the discussion, I would like to ask you a few quick rapid fire questions. The first one is one misconception about aging biology you would like most likely to correct?
Yeah, I feel like, and I myself included, I've studied like age-related disease. And I still, of course, believe in that type of work and that line of work and that research path. And I think if you're someone diagnosed with an age-related disease, you need people studying that and developing therapeutics. But I think that one thing that does need to change is that maybe we need to revisit how we're prioritizing these things. We haven't traditionally prioritized studying aging itself and longevity and health span, which like I said, are surrogate markers for, I think you can improve lifespan, then I think that is a good indication you're going to improve health span, which is what I'm after. And if you can improve health span, then you're going to improve age-related disease outcomes. And one way to tackle it is to look specifically at individual age-related diseases, but another way is to really look at aging itself. If you can slow aging itself, and if we were to, as a whole, kind of prioritize studying that, we may be able to improve age-associated disease outcomes more than if we just focus on individual diseases. I think we need both approaches, but I think more prioritization than traditionally has been given to the aging process itself needs to happen in terms of therapeutic development, right? We may have a better chance to improve human health by targeting aging itself versus targeting age-related diseases. So that's something I think fundamentally would like to see change in the aging field in general, in terms of kind of the research directions and the prioritization of research that's happening.
Excellent. Thank you so much. And the next one is one biomarker or process you think deserve more attention.
Gosh, I'm personally like, I study oxidative stress. So I think oxidative stress is always interesting because it's something that accumulates with age and it's a result of so many things, right? So if we can reduce oxidative stress or as a whole, I think there's, I don't know, I guess looking at things that reduce oxidative stress are likely to reduce all types of other aging hallmarks, my guess. But again, I study oxidative stress, so I think it's really important because it's not just something that can cause damage. It's oxidants or signaling molecules, and they play an important role in intracellular signaling and signaling to other cells. And so having this kind of appropriate level, you can't have not enough oxidants, and you can't have too much oxidative stress. You have that right balance of oxidant production and antioxidant responses. And maybe that's an area, I guess, that I think touches a lot of the other aging hallmarks.
Yeah, I agree. And I think that might be the cause of aging in a way, because all the rest might be the effect. I follow your...
Is increased oxidative stress a result of aging or the cause of aging? I don't think we know.
Yeah. So the next question is one habit people underestimate for long-term health.
Oh, geez. I think being social and I think there's something to be said about being socially happy, even at work. I'm fortunate that I get to work with just people I love to work with. I've been with Kosuke, who's the first author on the paper. His office is right next door to me here in Houston, and his office is right next door to me in Arizona and in Atlanta and here. And I'm just so grateful that I get to work with these amazing people that keep wanting to work with me. Yeah. So I think having this positive environment around you socially at work, I know sometimes for many people it's tougher to control their happiness at work, but I think it's important too, because you spend a lot of time at work. And so anything you can do to have a happy work environment is really important, and a happy social environment in general, I think is really important.
Yeah, makes a lot of sense. And to summarize, if the listeners would like to remember just one idea from your work on aging and your recent work and publication, what should it be? What should they remember?
Well, I think for the scientists, maybe to remember, just always be outside the box and thinking outside the box, trying new things, and never be scared to take risks. Sometimes people look at my career or my grants track record and they say, oh, wow, you've been so successful. And I said, let me just tell you, I fail 85% of the time, easily. So, just that failure is a part of the process, and failure brings you closer to success. So never be scared to take the risks and to fail, because that just takes you one step closer to the next big thing. And for people looking at the research, I think the main thing I want them to think about is that this is this new frontier, that psychedelics in aging, it's just never been studied before. And this really, I think, opens the door to who knows what's next. I'm really, like I said, I'm so excited to see what other people are doing as follow-up studies based on this initial study, and what comes out of this moving forward. And even looking at other psychedelics, I think take what their favorite molecules of interest in the psychedelics world and see if they have similar impacts. It's just something we've never studied before that seems to have a lot of promise, at least based on our study. So to be looking at this new area of psychedelics and aging, to be thinking about psychedelics in a way where we're not just always focused on what it's doing in the brain, but what else is it doing? I think that's probably the thing I want people to think about next, because that's what's going to lead to the next big breakthrough.
And yeah, maybe it's connected to your answer to the question about what people should do to live longer. And you said be social and happy. And that's in a way what psychedelic are doing, making you a bit more social and a bit more happy. So maybe that's the solution for the longevity problem. We don't know yet.
True, that is true.
Yeah, so that's an interesting connection. And Louise, thank you so much for this very exciting and new way of thinking. And I really hope that it will still be true in the future when a lot of other labs will look at that and test it. And I think that you open a new window for a new kind of research that I think that I'm sure that your lab will spend maybe decades on, but I hope that also other. And before we wrap up, I want to talk to the audience. If you enjoy Longevity by Design, please follow and leave review on Apple Podcasts, Spotify, and YouTube. It's helped others discover this science-first conversation. Thanks for tuning into Longevity by Design, produced by Inside Tracker. We'll see you next time. Again, Louise, that was an amazing conversation. I enjoyed it a lot, and I'm sure that our audience will enjoy it as well.
Great. Thanks so much for having me.
Thank you.
Thanks for listening to Longevity by Design. Please subscribe to this podcast on Apple, Spotify, or YouTube. Longevity by Design is powered by Inside Tracker, a personalized health optimization platform that helps people improve their lives by improving their bodies from the inside out, using personalized, science-backed recommendations for nutrition, supplements, and lifestyle changes. To learn more, visit insidetracker.com/podcast.